Hi. So we'll go ahead and get started on this Unit 3, Muscle Physiology. So you guys will notice this unit is 2 weeks, so you have a little bit more time, but the concepts in here are a little bit more complicated. So we'll start going through this here. So muscle can be categorized based into different groups depending on how you're doing it. So mainly there are three types. There's skeletal muscle, which is what we all think about. These are like your biceps, your facial muscles, anything that you have voluntary control over. Cardiac muscle is going to be in your heart. And then smooth muscle is going to be found surrounding your arteries, in your gastrointestinal system. So we can classify it based on those three types, or we can classify it based on striated or unstriated. And what striated means is striped. So when we look at it under a microscope, it has a striped appearance. Unstriated, smooth, looks nice and smooth under a microscope. Or we can categorize it based on voluntary or involuntary. So voluntary muscles are skeletal. You have control over your movement. And then involuntary muscles are the ones that we have no control over. So first we're going to go through skeletal muscle. Cardiac and smooth we'll cover later on in the unit. So muscle makes up about 40 to 50% of a healthy adult's body weight. We need it in order to have movement. We need movement in order to get energy. In terms of like food, we need movement for lots of other purposes. Okay? So muscles take ATP, which is chemical energy, and transform it into mechanical energy, which is movement. So these are a few root words for you to look up on your own time. Some functions of muscles, so obviously movement, walking, communication we kind of forget about, like in our facial expressions, in our writing. Muscles keep our posture nice, keep us upright. Muscles provide with protection and support, so like your abdominal muscles protect your abdominal organs. And then elimination of materials, so in your gastrointestinal system you have a lot of sphincters, which are little circular muscles that are typically closed, but they can open in order for us to eliminate any materials. And then like we talked about in Unit 1, muscle contraction releases heat and that helps us to stay warm. A few characteristics, so excitability. This just means that our muscles are able to respond to any type of signal. So the nervous system will signal the muscles and they are able to respond to that signal. Conductivity is electricity, so throughout the muscle cell membrane we can spread electricity. Contractility is the ability to shorten, extensibility is stretched, and then elasticity is just like a rubber band. So skeletal muscles, voluntary, striated muscle. These attach to bones primarily. So if we look here, this picture is a microscopic image looking at skeletal muscle. We can see the striations. You can see all alternating light pink, dark pink, light pink, dark pink. That is the striated appearance that you see on skeletal and cardiac muscle. And we'll go through why that is exactly later. So muscle has connective tissue that surrounds it. So on the very, very outside, you can see this layer is peeled back here. This is what we call the epimysium. So epi means outside, and this wraps around the entire muscle. The perimysium is around each fascicle. So this little group of muscles is called a fascicle. You can see there are multiple fascicles in the whole muscle belly here. So each little fascicle is surrounded with the perimysium. Peri means within. And then around each individual muscle fiber is what we call endomysium. So this is surrounding each individual muscle cell. So you might see muscle cells written as muscle fibers or cells. Same thing. And then we have other fascia that lie within the muscle. So you can see this is a cross section of a muscle. From the skin on the outside, then we have some superficial fascia, which is Mostly your hypodermis is where your fat is. And then the deep fascia is within a muscle belly in between each muscle. So 2 different connections here. A tendon is going to be a really thick cord-like structure that connects the muscle to the bone. And then an aponeurosis is going to be a really thin, flat piece of tissue here. These are basically the same thing. They're connecting muscle to bone, but tendons are thick and cord-like. Aponeuroses are flat. The muscular system is highly innervated with blood vessels and with nerves. So there are lots and lots of blood vessels within your muscles to allow for giving nutrients and removing waste from the muscle cells. And then we need nervous system communication in order to tell the muscle cells to contract or relax. So here is a little breakdown of the muscle anatomy. So this is a whole muscle here. And then if we look at just this part, this is what's called a fascicle. And then just this little part is a muscle cell or a muscle fiber, same thing. Within the muscle cells, there are groups called myofibrils. And within that, there are these little proteins called myofilaments. And you can see there's a thick one here that's big and a thin one here in blue that's smaller. And we'll go through each of those briefly in a little bit. So the way that muscles develop in embryonic development is they start initially as myoblasts. So myo means muscle, blast means build. These cells are building muscles. What happens is they fuse together, and then we end up with multiple different nuclei within one single muscle cell. Some cells do not fuse, and they end up what we call satellite cells. And satellite cells are adult stem cells, and they're there to be stimulated in the case of muscle injury. So once they get stimulated from injury, they will differentiate into skeletal muscle to help repair the muscle tissue. One single muscle fiber, one single muscle cell can be as long as 2.5 feet. This is your sartorius in the leg, which is a long, skinny muscle. Like I said previously, multiple mitochondria because the cells fuse together during embryonic development, and this is useful to produce lots of energy. You guys know mitochondria are the powerhouse of the cell, so they're the ones producing ATP, producing lots of energy. And then multiple myofibrils, which are these parts that include the thick and thin filaments, we'll go through in a little bit. And then multiple nuclei, again, from the fusion of multiple cells together. So here's a picture. You'll want to study this and reference it kind of throughout the semester, or throughout the unit, I'm sorry. So this is one single muscle cell or muscle fiber. You can see there's a plasma membrane here on the outside. This purple dot is the nucleus. We'll go through each of these different bands, but you can see like a Z disc or Z line. There's an A band and I band. These little things are little mitochondria. Plasma membrane is called the sarcolemma, and it has little extensions that go deep within the muscle here. You can see them in blue. This is called the triad. So this one single purple line is a transverse tubule, which is an extension of the sarcolemma, and then the blue on either side are the terminal cisternae. So these allow action potentials from the nervous system come into the cell membrane, and they travel and they go through the triad and go deep into the muscle here in order to signal the muscle cells to contract. The jelly inside of this cell is called sarcoplasm. In normal cells, this is cytoplasm. And otherwise, we'll go through each of these on their own, so don't panic. So these are the different components. We will go through each of them. So the sarcolemma is the plasma membrane of this cell. This is really important because it is able to conduct electricity signals from the nervous system. T-tubules are the continuations of the sarcolemma, and this is part of the triad that goes deep into the muscle, which allows the muscles to be signal. Myofibrils are these contractile units here. They're made out of myofilaments, the thick and the thin filaments. This is the bulk of muscle cells. So you'll have a bunch of these all running parallel to each other, and they'll extend the whole length. And we'll go through the different filaments later. Sarcoplasm is the cytoplasm of a muscle cell. You can see it here in like yellow. Sarcoplasmic reticulum, this one's really, really important. So this is kind of like endoplasmic reticulum. It stores calcium, which I guarantee will be an exam question. And what happens is once the nerves tell the sarcoplasmic reticulum to release its calcium, that is what will trigger muscle contraction, and we'll go through each of those steps. The terminal cisternae are these little green things on either side of the T-tubules, and they're part of the sarcoplasmic reticulum. So the triad, like I've discussed, is going to be 2 terminal cisternae in blue and 1 transverse tubule in purple. So these will connect the signal from the nerves and from the cell membrane into the sarcoplasmic reticulum. So this is a sarcomere. So you can see there are multiple. They go from a Z-line to a Z-line, these little zigzag parts, and then we have it blown up here so you can see this sarcomere. So this is the smallest part of the muscle that can contract, and you can see it even in a different cartoon here. So the sarcomere has light bands and dark bands. So we'll go through each of these components on their own. So sarcomere is Z-line to Z-line, these little zigzags. And this is the smallest part that's contracting. When the muscle contracts, these 2 Z-lines come closer together, and then when it relaxes, they spread back out to normal. So the I-band is just the small part here where there's just the dark blue lines. There's no of the pink lines. So this is only thin filaments. The A-band is just the thick filaments. So this is just the part with all the pink, the dark pink. There's an M-line right in the middle that anchors all of these to each other. And we call that the M-line, cleverly named for like middle line. The H-band is a small part within the thick filaments here that is just thick filaments, just the darker filaments. There are no thin filaments here. And then the M-line, like I said, is this thin protein in the center, and this is where the filaments attach to. So when a sarcomere is relaxed, like above, the Z-lines are far from each other. There's an H-band here with nothing in the middle. But then when it contracts, you can see the thin filaments, these little black lines, get really close together. They almost touch the M-line. The Z-lines get closer together, so the sarcomere is shrinking in size. They just overlap. So we'll go through the steps in a little bit. Oops. But I'll leave you at that for this first video.